Hello everyone. So let's start. So today is the last class of the cell culture technology NPTEL course, and so let's discuss about a little bit about the micro cantilever system and how the cells are grown there and how we can measure the force generated uh, the micro cantilever system MEA. Uh, so we'll talk something about that and then we'll solve some questions. So today there is not much to discuss, just a little bit introduction about the MEMS, and we will be then solving the questions, and if you have any doubts, we'll clear that as well. So let's begin. So so basically we will be talking about all the um, muscle cells and how the force generated by the contraction of the muscles can be measured using the microcantilever system. So how we can measure the force basically? So the force there is so all the muscle cells, basically the skeletal muscle, the cardiac muscle, smooth muscles, so these cells they contract because of the presence of actin and myosin. So these are the actin filaments, and this is the myosin filament. So this myosin has heads which can bind to the actin like this, and because of its binding to the actin, it can pull the actin together, pull the actin filaments together. So what will happen is the actin filaments will come close, and so the muscle will basically contract. And because of this muscle contraction and relaxation, muscles can perform different activities like the beating of the heart, or also the like skeletal muscle movements, smooth muscle contractions. So all of this is basically due to the activity of actin and myosin, the filaments which are present in this muscle cells. And this filaments basically work due to the activity of several background molecules like calcium signaling, sodium, potassium. So all these ions and their activity and their usage of ATP to they use up the ATP, and then they allow the actin myosin filaments to contract. So all this calcium signaling and sodium potassium channels, they get activated to allow this kind of uh, you know the muscle contraction to take place. So the force that is generated during this muscle contraction, that can be measured. How we can measure that in experimental conditions? So it can be measured using micro cantilever system. So cells are grown in this micro cantilever system, and we can use laser to detect the bending of cantilever, and we can basically see that how much the muscle is contracting or how much it is relaxing, so how much is the force. So what is micro cantilever? As the name suggests, micro, these are in micron dimension. It can measure molecules which are in the micrometer range. And so cantilevers are something, it looks like this. So this is basically a chip, and in this chip we can etch. So these are so this basically this surface is etched in the sense that there is a groove that can be made. So it looks like this. So each of this column, each of this micro cantilever, they are at a particular height from the surface. From this surface is at a particular height, as you can see in three-dimensional view. So this this allows that whenever there is cells grown on top of it, if these cells are contracting, or if the cells are relaxing, then this micro cantilever, due to because this basically these cantilevers, they are very very sensitive, and they can detect very like they can detect motions of motion, even if it is slight motion, that also it can detect. They are designed in such a way that they are very sensitive to motion, and they can they will not stay stagnant if there is any contraction or relaxation, and then it will bend. So here, as you can see, these are there in this chip. We can have many micro cantilevers, so many patterned. This is basically a patterned micro cantilever. This is a patterned cantilever, and we have an area where this particular like these columns are made, and these columns can be used for growing the cells. So once these cells are grown on the surface of this micro cantilever, we can expect that this cantilever, this is the cantilever before deflection. Like if there is no bending, then the cantilever is in straight position, so the angle between the base, so angle between the baseline and the cantilever is 90 degree. But if there is any force, if there is contraction and relaxation happening, then the micro cantilever will bend like this. So this is also the principle of the atomic force microscopy, or AFM. So AFM also detects the surface curvatures using this cantilever system. So these cantilevers look like this. So these cantilevers are somewhat like this, and they continuously tap on the surface. So whenever the surface is having groove, so in order to detect the surface groove, the cantilever will bend, and there is laser that shines on top of this cantilever. So depending upon how the angle of the laser bends, we can get an idea of how the surface is. Similarly, the micro cantilever system, where the cells are grown because of the force, here what happens is the cells are contracting on relaxing on top of it. So it will generate some force, and it will exert that force on the cantilever, and that cantilever will oscillate. So because of this oscillation, the angle of deflection, basically it will not be 90 degree. Here it was 90 degree initially, but after deflection, after generation of force, this angle will change from 90 degree to a particular angle. Like it will basically oscillate. It will come down, then again it will go up, again it will come down, then again it will go up. So because of this, it is going to get deflected, and this deflection we can measure it by measuring the angle. So we can measure this angle and then put it in Stoney's equation. So there is another mathematical equation where we can put this angle, and it will give us the approximate force that is being generated. So we'll not go much details into what is the mathematics part behind it, but the general principle of detecting the force generated by the smooth muscles is using this micro cantilever system, where cells are grown on the surface of the micro cantilever, and after their growth, this cantilever system, they will bend, because once the muscle cell attains its features, it is going to contract, and because of that, it will show deflection, and this deflection angle we can measure, and that angle we can use and put it into the Stoney's equation to get the force. Now this kind of method can be utilized in different ways of how the muscle contraction is affected by certain drugs. So if these are certain cells, and these cells, if they are adhered on the surface of the cantilever, and they are not showing any kind of deflection, or the muscle is not contracting, like some kind of diseased conditions where muscle contraction is affected, where the neurotransmitters are not released, and there is no muscle contraction. So maybe if we want the muscles to respond to certain drugs, then those diseased phenotype cells can be plated on the surface of this cantilever, and we can use certain drugs to incubate those cells with, and after certain time we can check whether or not these muscles are showing contraction by measuring the deflection of the cantilever. So the drugs can be screened based on the basis of how the muscle cells are contracting. So we can basically use microcantilever system to screen various drugs and check whether the activity of the muscle cells are being maintained or not. So how we can basically grow the cells? So we need to, so this is the micro cantilever system, and so in order to grow the cells only on this cantilever chips and not on the entire surface, people coat the entire surface with molecules which are, cytophobic. So this is the cytophobic surfaces, and so it is not going to allow the growth of any cells on the on the other surfaces. Whereas on the in the cantilevers, they will coat it with cell promoting factors, cell growth promoting factor. So cell growth promoting factors will be present on top of it, growth promoting factor, sorry. So cell growth promoting factor, it's going to allow the cells to grow only on the surface and will not allow the cells to grow on the other surfaces. So once we have done it, then we need to grow it in a particular medium, which medium is going to allow the growth of this particular muscle cells. So those medium can be standardized and optimized based on how which muscles we are working with and what phenotypes we want to study. So so we have to coat the top surfaces with cell growth promoting substances, and we have to coat the other surfaces with cell growth inhibiting substances or cytophobic area. And we can measure, so after growing the cells there, we can measure the deflection of the cantilever and use the Stoney's equation to measure the force that is generated by the myotubes. So the medium that is used generally for growing is a serum free medium, like the muscle cells need to be grown in serum free media, so that they can differentiate, and they will become the myotubes. And once they have done so, then we have to remove we have to add serums for growth of if we want to grow neuronal cells along with the muscular cells, we have to add a different set of media. So that is it, and we have to be careful, because as we were also discussing in the couple of classes in the last session last few couple of classes, that there are chances that we can get contaminating cells as well, cell types as well. So we have to be careful as to like what kind of medium we are using and what growth factors we will be using, because if we are not strict with it, then we will get a lot of contaminating glial cells along with the neuronal cells. Also even the muscular cells also, they are attached with other accessory cells that it interacts with. So we have to be careful as to what kind of medium we are using, and what factors we will be incorporating into that medium. And also, if you want to co-culture cells, if you want to co-culture the muscle cells and neuronal cells, what kind of medium should be common, so that both the cells can be grown without any problem, because both cells will have different requirements. And muscle cells will have different requirements, it will need differentiating agents. The neuronal cells will have different kinds of requirements. So it is very difficult as to like what kind of common medium can be suitable for both the type of cell. So over the period of time, there have been improvements in the co-culturing methods. So like generally we do not co-culture cells, because of one cell will always exhaust the nutrients that was meant for the other cell type. But during requirement we need to do it, and we have to be careful enough that not that only one type of cell should not be dominant in co-culturing conditions. All the type of cells should be equally present in order to see the required results or changes. So yeah, so we have to, then there are other like ways to which we can culture the neuromuscular junction on the microchip. So the neuromuscular junction, so neuromuscular junction is the junction where the neurons, basically this is the neuron, suppose, and this neurons is going to motor neurons, they will be having this their dendrites over here, and they will um, be attached to a particular muscle. So this muscle myotube, suppose, so this particular neurons, they are going to get some signals. Okay, so they will have some signals from the central nervous system or the peripheral nervous system, and they will want to convey this signal to the muscles and allow it to either contract or relax. So in order to contract or relax, so this particular muscle need to respond to the signal that is being transmitted by the neurons. So the neurons transmit the signals by various ways. One of the most common ways by the neurotransmitters. So we have neurotransmitters like acetylcholine, GABA, glutamate. So all these neurotransmitters, they work very differently. So GABA is an inhibitory neurotransmitter, so it inhibits the signal. Acetylcholine is an activating neurotransmitter, it will activate the signal, like it is not going to inhibit the signal. So there is the motor neuron, so there are different neurons also. There is the motor neurons, which are going to act on the muscle, and the muscle will respond to the changes or the signals, whereas the sensory neurons will sense the changes. So sensory neuron, they will take the information from the sense organs that we have, the skin, the eyes, ears, nose, everything, and then it will sense the changes, and then it will respond, and it will send the information through the motor neuron to the muscles for continuing their activity. So this neuromuscular junction, where the neurons are interacting with the muscles, this is the motor neurons. More specifically, they are interacting with the muscles to pass their signals. So this motor neuron and muscle junction is not very easy to replicate in the laboratory conditions, because they're the two very different cell types, a neuronal cell type and a muscular cell type. It is not very easy to co-culture them. Both are very difficult to culture. Neurons especially, they are very difficult to culture, as in like they will have adherence issue, they will not divide, they will not differentiate. So coming up with a solution where we can culture both the neurons and the muscle cells together, it's is in itself a very big thing. And so here the microfluidic chambers, this is one of the papers that I was seeing. So they had designed microfluidic chambers. So what they did is, there is two cantilever system over here. There is one cantilever and there is another cantilever. So each of this cantilever has different compartment. So one is a motor neuron compartment and another is a muscle compartment. So as to not if we are growing the cells in the same cantilever, it might be difficult, because there could be this dominance of a particular cell type, and it will not allow the other type of cell to grow, and the nutrients and exhaustion will also be there. So they have designed a very beautiful cantilever system, the microchip system. So MEA stands for micro electrode array. So this is a chip, so cell on chip method. This is a cell on chip method. So cell on chip, it is a very nowadays people are very much the modern cell culture techniques, they are following through this chip system. It's very handy, because you don't need a lot of media, or you don't need a lot of resources for this. But it is also tricky in the sense that we have to design the chip in such a way that it is going to be it is going to have the ECM. It is going to have a proper space for the cells to grow. It's not going to be like, it should not be it should not be an alienated thing. It should be very it should be very self growth friendly system. So here they have basically they generated a particular micro cantilever system where they have two chambers or two compartments, and they are culturing each of this cell type in a different compartment. So they'll have a motor neuron compartment where the motor neurons are cultured, and they have a muscle compartment where the muscle cells are cultured. So we have this neuronal cells. They will extend its processes. So these cantilever systems, they are very close to each other, they are not very very far. So this is one cantilever and this is the other cantilever, so it's very very close to each other, but it's not the same cantilever. So here they will be growing the neuronal cells, and here they are growing the muscle cells. So this is the muscle cells over here, and this is the neuronal cells over here. So they are growing the muscle cells and the neuronal cells, and the neuronal cells, once they get differentiated, they will send out their processes, and these processes will come and attached with the muscle cells. So here we can distinguish it, and it is to some extent it can basically like it will exclude the problem of the contamination chances of different cell types. This is a very good method for co-culturing, where two cantilevers are used for culturing the two different types of cells. So we have this kind of system. So in order to check whether or not the neuronal cells and the muscular cells are showing electrical activity, we can use some stimulus. So they can stimulate the muscle cells or the neuronal cells. Any kind of stimulus. For muscle cells, we can use some kind of neurotransmitters like glutamate or GABA or acetylcholine. So these are supposed to respond to those neurotransmitter and show contraction. Similarly, neuronal cells, they will have some electrical impulse. So if we are giving some impulse, then they will respond to that change. They will show some electrical activity. So motor neuron cells and the muscle compartment, their action potential and their the electrical stimuluses can be measured using again this, because what happens is this cantilever system, they are attached with electrodes. So they have a positive electrode and a negative electrode, so cathode and anode. This positive electrode and negative electrode, they basically can when we are stimulating it with any kind of impulse, so it is, if any kind of movement is there in the cantilever, this kind of electrodes can sense it, and they will show a spike, or the we can get a graph which will show an electron, which will show an electrical movement, so an electrical activity. So the electrical activity of neurons can also be observed, and the electrical activity of muscle can also be observed here. So they confirm that the neuronal cells and the muscular cells, they are growing together, and they are showing the electrical activity. And also, along with the electrical activity, there should be some mechanical motion of the muscle cells as well, because they are going to show contraction. So this contraction, this mechanical activity can also be measured using the using this cantilever system also, because they have, I think, some kind of way to measure it, the mechanical movement of the muscle cells. If it is contracting, it's going to move a particular direction. It is going to, again, if it's relaxing, again it's going to it's return to its original position. So all those mechanical forces and the mechanical motion can also be detected. So the neuromuscular compartment, the neuromuscular junction, can be replicated in the microchip system, and in this way we can basically be more closer to the physiological condition of how the neuromuscular junction, it is so how the neuromuscular junction basically is present in our body in the physiological condition. We can replicate it using the two chamber system, growing the different kind of cells. So once we have cultured the cells, and once we are having the neuromuscular junction that is formed, then we can continue with our experiments with like stimulating the drugs, stimulating the neurons, and then checking what the muscle activity, or inhibiting the muscle activity and checking whether the neurons are working, checking the drugs. So this, all this system basically is like modification, like whatever cell culture we are doing, the normal cell culture where we are plating the cells in dishes and trying to use different drugs, different stimulus to see what happens to the cells. So these are like more different from physiological condition, but more and more how the science is progressing, the cell culture technology is also progressing, and the cell culture is becoming more and more physiologically relevant. So it is much more reliable result, because we will be mimicking the entire neuromuscular junction, the entire cellular activity which is present in the physiological condition. So the micro cantilever system and the microchip system, it's very like advanced level of cell culture, and definitely it is going to be like in the future, definitely is going to be used in more amounts. Now also it's not that like that common, as in like these are very expensive chips actually, so it's not very handy and very economical. So there are labs, even in our even if in our institute as well, those who work with the microchip system. So it's very, I mean, expensive and very we have to maintain it, because these chips, they are so small you cannot see it with your naked eyes, and they can get easily contaminated with dust, or they can easily be contaminated with other bacterial cells. So this kind of chips, even when they are making it, they have to be made in a sterilized condition in every, like I mean, hood and all those things. So those who work with this chip system, they have to be really careful with that as well. So the co-culturing of neurons and muscle cells, so basically we have to plate the muscle cells first. So if we are plating the muscle cells, and then we can use the differentiating and the cell division factors as well, because the muscle cells, they initially need to divide, because the muscle cells initially will, these are the singular muscle cells, then they will divide like this. So once the muscle cells divide, and then once they are divided, then they will join together. So all this line basically will be erased, and we will get one singular tube like of structure with identical, there will be identical nucleus. So this nucleus, they will not fuse with each other, they will have their own identity. And so uh, so this is, we have to add FGF and then leave for, this is not lever, this is leave for 2 to 3 hours, and then so this will basically allow the cell division and differentiation. And so muscle cells will basically they will divide and differentiate, and they will form myotubes. So once they are forming the myotubes, the muscles are bound to show the activity of myotubes. It should contract. So then we have to remove the media which contains FGF. So we have to remove the media partially, so as to not dry the cells completely. And so the media should be only covering the top part of the cells, only the top part of the cells, and the rest of the media. So basically we are having the cells which are plated like this, and the media was till here with FGF, and so it will allow the differentiation and division of the muscle cells. So once the muscle cells reach the state of myotubes, which will take around 2 to 3 hours, the rest of the media is removed, and only till the the top portion of the cells, just an estimate how much media we can leave. So just the top portion of the cell should be submerged, rest all the media should be removed. Now, after removing this media, we have to add media which is not having any FGF, because we don't want any more cell division. Now the cell differentiation is going to happen from motor neurons. So the motor neurons, basically they are isolated from the embryonic stage, the embryonic 14 from the ventral horn of the rat embryo, and after 6 to 10 days, the neuromuscular junction forms. So we are using different types of media for culturing the muscle cells and for culturing the kind of the neuronal cells. And so after after adding these motor neurons, and after allowing it to grow for 6 to 10 days, there should be the formation of neuromuscular junction. And this neuromuscular junction should be activated, and that we can check by either adding glutamate to the culture. So glutamate is going to show the activity of so glutamate again is a neurotransmitter, and it's added on the dish. So it will, this glutamate, it's a neurotransmitter, and it will. So if we are having the neurons plated with the muscle cells, and we are adding the glutamate, and we are seeing there is changes in the muscle, that means that the glutamate is actually showing its effect as a neurotransmitter, and so the neuromuscular junction is properly has to be properly formed for showing that effect. Also, we need to check by stimulating the neurons and then checking whether the muscles are showing the contraction or relaxation. So all those experiments need to be performed so as to confirm whether the neuromuscular junction is formed correctly. Also, apart from adding glutamate, another thing that they have to add is the glial cell. So this glial cell, basically it is present just adjacent to the neuronal cells. So these are the neuronal cells, and glial cells are added on the side, and this acts as a nutrient media, like nutrient reservoir, and it will give nutrient to the neuronal cells. And apart from that, it also absorbs the neurotransmitter, and it will allow the neurons to pass the information very nicely to the muscle cells. So addition of glial cells also increases the electrical excitability of these neurons. So so this by adding the glial cells and by adding glutamate, we can be sure whether the neuromuscular junction is forming. Also, another way to you know check whether the neuromuscular junction is forming is by using antibody against the acetylcholine receptor. So antibody against acetylcholine receptor. So these acetylcholine receptors are present very high amount in the neuromuscular junctions, because they are going to transmit the signal to this neurotransmitter. And so if there is a if the antibody binds to the acetylcholine receptors, then it's probably it's probably the neuromuscular junction is formed. So there is not one way to confirm neurovascular junction formation. We have to confirm it using different methods, so that we cannot rely on one method, as because already the technique of microchip and this cantilever system is very new. So everything that we are doing, we need to, not only just for this, every other experiment as well, we have to be sure enough that whatever result that we are getting is confirmatory, because we cannot rely on just one experiment. So like people will do different experiments just to prove the one thing. Different experiments to prove only one thing that is correct. So definitely, so for the formation of neuromuscular junction, because everything from here again relies on whether or not the neuromuscular junction has been formed correctly. Because if it is not formed correctly, and if we have not tested it, and we are seeing some drug and we are not seeing any changes in the contraction level or any changes in the cantilever deflection, we will not be sure whether it is due to the effect of the drug, whether the drug is showing no effect, or whether it is the neuromuscular junction that has not formed properly. So it is we have to be sure enough that whatever we are doing is like reliable, and it is also physiologically relevant. So then we have to like, as we are as I was saying, that neuromuscular junction, we can use specific antibodies against the acetylcholine receptor. to check whether or not the neuromuscular junction is formed. And then we can also, apart from that, neurons should express other kinds of channels like sodium channels, potassium channels, and sodium ATPase pumps, the sodium potassium ATPase pumps. So all these channels, they are very important, as in their activity, the ions activity is very important for uh, causing the muscle contraction. So if this ions are not properly released, or channels are not properly working, then the muscle contraction will not happen, and the muscle will not show the required contraction. So this expression of sodium channels, potassium channels, sodium potassium ATPase pumps can also be seen with the help of antibodies. So these neurons ideally should express all these channels properly in ideal amount in order to show the electrical activity. And this electrical activity of the neurons and muscle cells, they can be used, they can be measured using electrodes to measure the spikes, as here, as it is shown here. So electrodes are used to measure the electrical spikes. Like if we are adding any stimulus, then it should be able to generate an action potential. So this action potential basically is the electrical impulse that is generated on response to a particular stimulus. So So electrical excitability of the neurons and the muscle cells can be measured using those electrodes. And so we have to be, different, and there are different kinds of muscle cells, the different kinds of neuronal cells also. So these are glutamate, so they will release glutamate, and acetylcholine, ACH, and then GABA. So all these kinds of neurotransmitters, they behave in a different way, and the neurons which are secreting this kind of neurotransmitters are also different. So not all the neurons will secrete a single type of neuronal transmitters. So depending upon what kind of neuronal transmitter it is, we can use different stimulus. So all this can act as a proper stimulus. So GABA is an inhibitory neuron, so it will inhibit, it shows inhibitory stimulus. Like if the stimulus is there, then the muscle is not going to contract, or if the stimulus is not there, it's going to contract, like that. So uh, totally opposite result of adding GABA. So that is all for micro cantilever system, and how we are, culturing the cells of neuromuscular junction. So today there was not like much to discuss, because this was kind of very advanced kind of cell culture technologies. And like this is still a very very new field of exploring. And there are like, yeah, as I said, there are labs that work on the chips, and these are kind of still very new and very, there is a lot of things to explore here, and there's a lot of things to optimize as well. So yeah, so that was it from the course. And so we'll move into questions. So we will not, if there is any other questions or doubts, we can solve them, and otherwise we'll move into questions. So yeah, so we can move into the questions. Okay, so Vijay has given the correct answer. So yeah, so the term histotypic culture, it implies that the cells which are grown in three-dimensional tissue-like structure. So the propagated cell lines, basically all the cell lines, if they are grown in a three-dimensional matrix, so all the three-dimensional matrix which is suitable for growing the cells and has the required nutrients. So those are the three-dimensional tissue like structure. It will show three-dimensional tissue-like structure, and that is termed as histotypic culture. Next question. Okay, so the answer for this is option D. So macrophages are actually adherent cells only in the blood. So in the body, it shows phagocytosis, and it will so it moves very slowly because it's not going to roll. It's going to get on the surface of the blood vessels, and then it will roll on its surface and will find out where the foreign substances are, and then there's going to phagocytose it. So it's our adherent cells, but when they are grown in culture, so when they are basically like when these macrophages are isolated and then they're grown in culturing conditions, they seem to be not fully adherent. They are semi-adherent. They are floating cells, but in the in vivo system inside the body they are adherent. But in the culturing, when they're cultured outside, so that time they become kind of non-adherent. So they are inherently adherent. They have inherently adherent properties, but in culturing conditions they turn out to be non-adherent. So the correct answer is, oh, sorry. The answer is non-adherent. This is not D. This is not. Okay, so uh, this is sorry. Macrophages basically they are non-adherent only. But somewhere I was reading that it was in the this inside the body they are adherent cells, and when they are outside the outside the system they are non-adherent. One second, I will also check this answer again, because here I have written it option A, but as far as I remember, it was adherent. So I will check it and I will again let you know. Uh, but yeah, so let's move on to next question. Yeah, Vijay, I think I have somehow not seen the correct answer, because I also remember it being adherent. So this question I'm having a little doubt. I will check once. Yeah, this is actually adherent only. The option is here. It is not A. It is D. Actually, it is adherent. So inside the body, these are adherent cells only. But after they're, after they are seeded, that time they become non-adherent. So ignore this option. I will change here. The answer is actually option D. It is adherent. Okay. So this is the correct answer. Option. Next question. Okay, so yeah, so the correct answer is organoid. So the definition obviously here, so organoid basically it's a three-dimensional cell culture. Okay, so it incorporates most of the features that are present in that organ. So this cells are taken from the organ of the respective organ. Suppose if you want to represent a kidney, so the cells will be taken from the kidney and then it will be grown in 3D culture. Different types of cells they will be grown in the 3D culture and will show characteristics of that represented organ. So these in vitro culture systems, these are characterized by the self-organization of multiple organ-specific cell types into a spatial organization which is similar to what is observed in vivo. So in vivo conditions, we have different types of cell types which are making the particular organ. So different tissues are making the organ, and so the cells are also grown in such a way that all these cell types are arranged in an organization that can be that can show similar kinds of features, key features of that representative organ, and so that is why the name organoid. And it is also capable of recapitulating some of the functions also as well of the Yeah, so the correct answer for this is option D. So for embryonic stem cell biology, the protocols are always, designed in such a way that can improve all the division, differentiation, as well as survival of the cells. So the correct option is option D. So next question. So yeah, so the correct answer for this is option D. So this is CNS stem cells. So stem cells, they are showing the self-renewing capacity. Also, they are proliferative and they are multipotent because they can differentiate into different cellular types like neuroectodermal. Also includes different neuronal and glial subtypes as well. So the correct option is option D. The next question. Yes, so the correct answer for this, so encourage independent cell culture is for the hematopoietic cell culture. So correct. All other cell types like cardiac or skeletal or neuronal, they are anchorage dependent and they require substratum or the ECM or the matrix, extracellular matrix for growing them, like they are adherent cells, and hematopoietic stem cells or the hematopoietic cells, these are anchorage independent. So the right option is option A. The next question. ES stands for embryonic stem cells. Okay. So yes, so the correct answer for this is option D, the embryonic stem cells. So these are derived from the isolated inner cell mass of the blastocyst stage embryos. So other cells, these are not derived from the blastocyst stage embryo, the inner cell mass. So the inner cell mass gives the embryonic stem cells. So the correct option is option D. So with that, that is the end for this course. And so yeah, so that is all for today's class. So I'm finishing off early. So that was like I had this many questions only, like I could gather. So you have any other questions or doubts? We can discuss, and then. Uh, so yeah, really it was very nice course. Very knowledgeable for me as well. I had a lot of fun teaching it, and I learned a lot of things. So thank you for everyone for joining. I hope you also learned along with me. So, you have any questions?